Thank you all for joining our session today. So I'm Seung-Il Lee, Assistant Professor at the Lakehead University, Thunder Bay, Ontario. I'm trying to moderate this session and will do my best. So our session is post-disturbance recovery of forest biodiversity after harvesting and natural disturbances. And we'll be covering from insects to bryophyte plants, ecosystem function, and policy. And I'm sure it'll be a really exciting session today, and I hope everyone will bring some inspiration and research idea through the sessions. So our first speakers are Anne McIntosh and Selena Schutt. Dr. McIntosh is an associate professor at the science department at the University of Alberta, and Selena Schutt is research assistant in the same department. So they are going to talk about post-harvest biodiversity recovery after variable retention harvesting in boreal forest. So please welcome to Dr. McIntosh and Selena Schutt.
I don't need to introduce myself further because Seung-Il has done a great job of that, except to say that the Augustana campus where I work is in Camrose on Treaty 6 territory. And I'm actually currently at the University of Victoria on the traditional territory of Legacies. So I'll leave it to Selena's going to do the first half of our presentation, and then I'll do the second half.
I'm speaking to you today from Airdrie, Alberta, home of the Treaty 7 territory, containing the Blackfoot Confederacy, Tsuut'ina First Nation, Stoney Nakoda, and the Métis Nation of Alberta in Region 3. Like Seung-Il said, we'll be talking to you today about post-harvest biodiversity recovery after variable retention harvesting in a boreal forest. As soon as I make the slides come, there we go. So, study background. So, our research focused on boreal forests. And boreal forests are dynamic ecosystems, as they are both natural and they can be affected by natural and anthropogenic disturbances. And it's important to note that there is added complexity in anthropogenic disturbances, as they also change over time. The landscape naturally includes a variety of forest ages. And so our big overarching question was, how long post-harvest does it take for biodiversity recovery in boreal forests? We speculate that recovery is likely not linear or even clearly directional, but hopefully returns to pre-disturbance scenarios. In order to determine the recovery time, we need to characterize the natural variation in the reference. So the objective is to quantify ecological recovery towards a mature forest of the biodiversity in boreal forests in response to varying degrees of forest harvest. So that covers everything from clear cuts to 75% retention. However, beyond the Nmds study that I'll talk about in a minute, the focus is less than 20% retention. And we've examined deciduous mixedwood and coniferous forests from across the world, not just at the Nmds study in Alberta. So our methods were first to identify data sets. We've sampled or did a systematic review of over 450 papers, which includes the Nmds data. From there, we identified taxa of interest and came up with 6. Understory plants, both vascular and non-vascular, lichens, birds, small mammals, and arthropods collectively and separated into beetles, spiders, and other. I next contacted researchers with the help of a former research assistant, and we narrowed the 487 down to 264 articles. Next was formatting data sets. Of the ones we did receive, there were 82. From there, analysis was run in our statistical software, using first a Chow-Jacquard similarity index, then a linear regression, and then a generalized additive mix model For the EMEN data, it was a GAM, and for the compiled data sets, including all the compiled studies and EMEN, it was a GAMM. So the EMEN experiment that I've been talking about stands for Ecosystem Management Emulating Natural Disturbance. It includes a bunch of studies conducted in a forest in northwestern Alberta. And the age that we have studied so far is up to 16 years post-harvest for our study. It covers everything from deciduous dominated to conifer dominated stands with total mixed wood, as well as deciduous dominated with conifer understory forests. It also covers everything from clear cut to 75% retention in stands and mature forests that may or may not have previously been harvested, but have returned to a mostly natural state. And with that, I will turn it to Anne.
Excellent. Thank you, Selena. So yes, in this study here today in our presentation, rather, we're just going to be focusing on ground beetles or caribids. So we're interested in showing you how beetle community diversity has recovered after forest harvest in boreal forests. So we've got 3 result slides that we're going to show to you. We've got 2 focused on the EMEN data, and then a 3rd that is just like a sneak peek of coming attractions ran freshly analyzed this week by Selena, our larger, broader data sets from the systematic review for the caribids. Next slide, please. So I'll just walk you through, if we draw our attention first to the graph in the upper left, all of the graphs on the following the next 3 slides, you're going to see the same axes, but different time periods and different types of forests and retention levels. So draw your attention first to the y-axis where we have the Chow-Jacquard similarity. So that's the similarity of the community composition, ranging from a value of 0, where there's no similarity at all, up to 1, where there's 100% identical composition in the caribids amongst sites. So, and then on the x-axis, you have the years since harvest. In this case here with EMEN, we're looking at 1, 2, 6, 11, and 16 years post-harvest. And you'll see that here we're looking at the 4 different forest types that Selena mentioned in the previous slide. So this is just for the EMEN data. You'll also note that we have a p-value there, all that are significant at a value of less than 0.001. And that's just telling us that these lines, that there is a relationship here. So it's not just that there is no pattern over time. So looking at the Gam model results here, you'll look at the shape of the curve. And what I want you to draw your attention to is on the right-hand side of the graphs, you'll see a little red point with a 95% confidence interval around it that Selena will highlight for you there. And that is the suite of values, the mean value with the 95% confidence interval for our reference condition. So that's pooling all of the reference data together. And so what we're doing is we're looking at our curve with the 95% confidence interval for our blue line around it over time. And what you'll see is for the coniferous dominated forest in the upper left, that we don't have recovery after the 16 years. And we see the same pattern if we drop down to the lower right graph, similar shape in the mixed wood forest. And in contrast, and when we look at the deciduous dominated, whether it's just deciduous dominated or with the conifer understory, so the upper right or the lower left graph there, what you'll see is that we see an initial decrease in the caribid diversity and then recovery. So that's because at age 16, our blue line and the confidence interval overlap with that reference red dot with the confidence intervals around it. So we see differences amongst the treatments and recovery just in the deciduous forests. Next slide, please. Okay. So what we're looking at now then, so we're still at the EMEN site here, so still at our smaller time period, our 1 to 16 years post-harvest. And now we're just looking at the coniferous forests. So in the last slide, we looked at clear cuts for all 4 of those different forest types. And now what we're doing is looking at a single forest type, the coniferous forest type, with no retention, all the way up to 75% of the forest retained. So we know that this is sort of more extreme ranges of retention that you would expect to see under traditional silviculture harvest, but that's part of the design of the EMEN to see what's happening there. And what you note is you see a consistent pattern across all levels of retention. So our clear cut, we see the dip and no sign of recovery. And then we see the similar pattern with the 10% retention. And then when we go down to the bottom graphs, then we can see we've got 20% retention, 50% retention, and 75% retention. And in fact, interestingly, I think, at least it was to us, is looking at the 75% retention level. That's actually like a linear relationship and going, away from being recovered. So that was an interesting finding for us. Next slide, please. So then in our last result slide here, this is the one, the fresh breaking, hot off the R studio analysis we have here. Again, our chow jacquard similarity, but now what we're doing is pooling all of the data sets combined. So for caribids, 19 of the data sets that we had were caribid. And you can see that 13 of those were for coniferous dominated forests, 2 for deciduous dominated forests, and then 4 for mixed wood. And again, because these are different data sets, we have different time periods since harvest. So we'll start by looking at the coniferous dominated in the upper left. And what we see here is that we hadn't seen recovery in the Nmds sites in the time period under which that was analyzed. But what we do see is around the 35-year mark, where we do see the recovery having occurred. So it's just that it takes longer in those coniferous forests compared to the deciduous forests, because now when we pool the Nmds data set with our other data set that had caribids, we can see that the recovery basically, like, there was a slight decrease, but overall recovery was consistent throughout the time periods. And in contrast, if we look at the bottom graph, the mixed wood forest, what we see there is that there is a decrease over time and no sign of recovery. But this is just capturing the first 20 years. So if we had additional data sets going past that, we could better address at what point recovery happens, because we're not there yet. Next slide, please. So overall, at this stage, I know we just sort of hit you with a few graphs there, and we've just focused on the caribids, but just to reiterate recap what we've just said, we can see that the rate of recovery is definitely varying among the stand types for the caribid beetle communities. It appears that the deciduous are recovering more quickly than the coniferous forest. We're not quite sure what's happening yet with the mixed wood forest. And doesn't appear to be that if we look at Nmds or those extreme levels of retention, doesn't seem that there's any kind of threshold there for a higher level of retention leading to resistance of the caribid community. So that was really interesting. And so we'll have to stay tuned to see with mixed wood forests if we get more, in the future when there is more data for older time periods to better answer the questions about recovery for that. And overall, the study that we're doing here, definitely there's huge value in long-term data collection, not just one-off data sets that we're working with, but I think we've really appreciated the value of Nmds, and we're excited to have other studies similar to Nmds that follow over long time periods and see what they find, and the repeated measures over time gives us really robust, valuable data. And we're excited for our data to provide insights into sustainable forest management and help inform biodiversity management framework, which is something that's really important in Alberta, where we're based. And John Statt's going to close out this symposia highlighting and talking more about that. And so I'm sure you're anxious to hear more about all the other taxa that we focused on as well. So stay tuned for more information as we wrap up this project later this fall. So next slide. So before we wrap up, we want to acknowledge our former research assistant, Michelle Rude, who worked on this project, all the researchers who've shared their data set, all the researchers and the field assistants at Nmds and associated with all these other data. And we're very grateful for the funding that has supported first Sangil and then Selena on this project, including the Alberta Biodiversity Monitoring Institute, the Alberta Conservation Association, and MyTax. And then there's the other funding partners down below that have funded the Nmds project specifically. Thank you for your time, and we're happy to take questions.
My name is Terry Porter, and I'm a molecular ecologist with a background in mycology. and I'm currently working in the field of biodiversity genomics. Today, I'd like to talk about some work I did with Natural Resources Canada at the Great Lakes Forestry Center with my co-authors, Emily Smenderovac, Dave Morris, and Lisa Vinier. The title of our talk is The Road to Recovery Following Wildfire and Harvest Disturbance. So Canada's boreal zone spans about 5.2 million square kilometers, shown here in green. And in this area, wildfire is a natural stand replacing disturbance, shown here by the brown points. Jack pine is a very common boreal forest species that is commercially important, and there are many fire origin jack pine stands at various stages of establishment in Canada. So in forest management practices that emulate natural disturbance, a stand replacing wildfire is often treated as the reference condition when assessing recovery following harvest, and that is the approach that we take here as well. So in this study, we're using a technique called DNA Metabar Coding, and this is an approach that's gained traction in government programs such as in the Ecobiomics project, and I've included a screenshot of the publication that describes that Government of Canada program here. And this approach basically involves going out and collecting environmental DNA samples, such as bulk soil cores that have been homogenized. And from that, you can extract whole community DNA without having to isolate or identify individuals in the sample. And then these are processed at scale, sequenced, and taxonomically assigned with a measure of confidence using various bioinformatic methods. So using this approach, we have chosen to focus on the recovery of soil communities following disturbance. So in this project, our first objective was to establish a temporal baseline of recovery following natural wildfire. And we did this using a number of different sort of lenses, but all I have time to talk about today is the recovery in terms of beta diversity, and that will become our reference condition. And I will use that to then assess the recovery of soil communities after clear-cut harvest, and also look at the impact of harvest type on these soil communities. Our samples were collected from jack pine dominated boreal forest in northwestern Ontario. And the table at the bottom summarizes the number of sites that were sampled in triplicate across 5 different age classes and 3 different disturbance types. So wildfire is our reference condition. And then we also had sites that were clear-cut harvest and salvage harvest. This is an overview of the 85-year chronosequence following natural stand replacing wildfires. And what I'm going to show you next are basically the recovery trajectories of various components of the soil community that were sampled at each of these age classes. So we used permutational analysis of variance, PermaNova, and we found that a stand development stage explains 32 to 45% of the variation in these communities. And I'm visualizing these community shifts using non-metric multidimensional scaling, NMDS. And I've colored the points and sort of shaded the groupings according to age class. So the lighter colors represent earlier, younger age classes, and the darker colors represent the older age classes. And what we find in general is that the younger age classes are associated with higher pH and higher temperature, and the older age classes are associated with higher total organic carbon and higher moisture. And when we use pairwise PermaNova to compare these groupings, we do find that that age class explains a significant amount of that variation in beta diversity. Now, another way to assess recovery and community stability is to conduct whole community nestedness analyses. And I went about this by calculating NAFEW. It stands for Nestedness based on overlap and decreasing fill. And I've added the citation to this method here. They explain it very clearly. But here I'd like to show you how to apply that to our data, which is DNA Metabar Coding data, and how you can interpret that. So in the bottom left corner, I've broken out the results according to soil community group. This is for soil bacteria. And each row represents an age class. The age class with the highest richness is always the top row, and the age class with the lowest richness is always the bottom row. And so when you read this plot from top to bottom, it's telling you something about the nestedness of the communities at each stand development stage. And wherever you see a gray bar, that indicates a presence, and wherever you see a white bar, that indicates an absence of a sequence variant. And so NAFEW tells you something about this nestedness structure top down, but it also tells you about occupancy if you read the graph from left to right. So you can see that all the bars are pushed to the left. This grouping here, this large gray rectangle, basically represents the core bacterial community where these sequence variants were detected at each age class. And then towards the right, you see which sequence variants are sort of popping in and out from one age class to the next. Now, if we just compare this large core community for bacteria in soil with the core communities of fungi and arthropods in soil, what I hope you notice right away is that the relative size of these core communities are much smaller. And what that indicates to us is that the fungal and arthropod communities in soil are sort of undergoing this dynamic reshaping from one age class to the next. Now, I wish I had more time to talk about community assembly processes and what else you could get out of this. I don't have time for that right now, but I'd be happy to answer further questions later. I chose to include these though, because I think these fingerprints really make it a visual way to show which variants are popping in and out and which are conserved across age classes. And so that was sort of establishing our reference condition. And moving on from there, what I'd like to do now is then look at the trajectory of recovery from clear-cut harvest sites and then compare that to our reference condition. And so this slide is just an overview of the type of comparisons that we made to look for convergence. So if we break it down one taxon group at a time, we're looking at soil bacteria first. I'm showing Nmds plots, they're color-coded. So the reference condition is always shown in red and clear-cut harvest communities are always shown in blue. Each column represents an age class. So E for establishment, crown closure, onset of self-thinning and late self-thinning. And if you see an asterisk, it indicates that we ran a permanova and found that disturbance type explained a significant amount of variation in beta diversity. And so the conclusions I'm drawing from these series of plots is that the bacterial community as a whole begins to converge at about the crown closure stage. But we do see different patterns for different components of the soil community. So when we look at fungi, you'll notice that each of these communities appear distinct at each stage compared with the reference condition. And the arthropod communities, they do converge, but not until the late self-thinning stage. Now, we didn't just look for convergence. We also wanted to see that if given more time, would these clear-cut harvest disturbance soil communities approach the reference condition at the mature stage? And so that's the next set of comparisons that we made. And so here, what I'm showing is more Nmds plots broken down by soil community. And the reference condition is always shown in red, and in blue are the various stages of clear-cut recovery communities. The lighter colors are early stages, the darker blues are the later stages. And for fungi and arthropods in particular, you can really see the direction of recovery towards the reference condition. But when we run our pairwise permanovas, we find that the age class still explains a significant amount of that variation. So when we start to then look at the impact of harvest type, what we compared was recovery following clear cut and recovery following salvage harvest. And we compared that to our reference condition at the two age classes for which we had good data. And so I'm showing more Nmds plots. They're color coded. So the red represents the reference condition. The blue represents clear-cut harvest sites, and the brown represents salvage harvest sites. And at the establishment stage, we do find that these communities cluster together and they are distinct. But when we move on to the crown closure community, we do start to see convergence of the clear-cut harvest communities approaching the reference condition, but we do not see that for the salvage sites. And when we test that using pairwise permanova, we find that the only communities that seem to have converged are the bacterial communities at the clear-cut harvest sites. So the key takeaways from this presentation then are that we created this wildfire baseline of recovery, and we found that each age class harbors a unique soil community during its recovery following wildfire. And what that would mean in terms of forest management is that preserving a mosaic ecosystem that represents each of these stand development stages is needed to maintain the full suite of biodiversity. When we look specifically at the recovery of these soil communities following clear-cut harvest, what we find is taxon-specific convergence. And so the bacteria tend to converge most quickly at the crown closure stage. The arthropods tend to converge with the reference condition at the late self-thinning stage, but the fungal communities and the mycorrhizal communities are quite distinct at each of those stand development stages. And when we check against the reference condition, have they approached the mature community following wildfire, we find that they are still distinct. They have not yet fully recovered, but our plots clearly showed that the trend is in that direction. And what that means for forest management is that clear-cut harvest communities may need at least 64 to 90-year rotations to allow the full suite of biodiversity to recover between clear-cut harvests. And when we look at the impact of harvest type, we find that clear-cut harvest soil communities do converge or approach convergence by crown closure, but the salvage harvest communities do remain quite distinct at crown closure. And what this means in the future is that we may need to limit the spatial extent of salvage harvest following severe natural disturbances. And all of these results are consistent with what we already know in the literature based on the quicker recovery of bacteria in soils, and bacteria also seem to be more resistant to heat and fire compared to fungi, or at least recover more quickly. And we do know also from the literature that there are things we can do to perhaps facilitate the recovery of harvested communities, including keeping coarse woody debris and perhaps setting up green tree retention rule sets that would preserve habitat for soil fungi and arthropods. And that wraps up my presentation. So thank you very much for your attention. I'd like to acknowledge funding from the GRDI Ecobiomics Project, support from the Hajababai Lab at the University of Guelph, and I've included my contact info in case anyone has any further questions later. Thank you.
So thank you for the introduction. It will be my first presentation in English, so I hope you will understand me. So I'm happy to present you a part of my research thesis, which talk about the bryophyte community's response 20 years after forest management in boreal mixed forest. So with Maxence Souberan, Timothy Work, and Nicole Fetton. So I suppose you all know that forests are dynamic ecosystems which evolve over time. And in boreal mixed forests, we can schematize these dynamics with the 3 different states by looking at the structure and composition of the stands. So we have the initial state, so young forests with homogeneous structure, composed mainly by hardwood species, and then the mixed succession state, so more mixed with the composition, and then the oldest forest with a more coniferous species. So stand dynamics represents the transition from a structure to another, and in boreal mixed forests, we know that natural perturbation like fire make change the structure. So fire is considered as a primary disturbance, and you make return stands to more homogeneous young forest structure. And with insect outbreaks considered as a secondary disturbance, they maintain a stand and sometimes make them accelerate their transition to a more mature stage. So this natural dynamic and these 3 states represent the principle of the 3 cohorts. And so there are the 3 states that will serve as a reference to practice the ecosystem forest management. So this is a management approach that aims to maintain a healthy and resilient ecosystem by focusing on trying to narrowing the gap between the natural and the harvesting stands. And we try to do that by varying the different harvesting technique to preserve this mosaic of stands that we observe in the natural landscape. So we have the clear cuts. We will bring back the stand to more homogeneous and young structure, and the partial cut We will preserve the stand in the initial state, or even sometimes, we hope, make them accelerate their transition to more advanced stage. So we know this well for the trees, which constitute a big part of the forest, but we still have to develop our knowledge on what happens below at smaller scales. And that's why bryophytes constitute an excellent subject to look. So they are a really important component of forest biodiversity, and they play a lot of ecosystem role, such as carbon concentration, water filtration, or again, soil thermoregulation. And so their unique physiological and habitat requirements make them really sensitive to forestry operation, and thus they are a really good indicator to study for the consequences of harvesting on the biodiversity. So this is why here we use them to know more about the influence of different harvest intensity 20 years after application. So how we did that? Our study area is situated in Quebec in the SAFE project. So it is in the mixed wood zone of the boreal forest, and it's situated in the Lake du Parquet Research and Teaching Forest in the Abitibi region. So SAFE, it's the French acronym for Sylviculture et Aménagement Forestier Écosystémique. And the project was established 20 years ago. And it constitutes a series of silvicultural experiments which were carried out in the 3 stand steps. So the young stand, medium stand, and old stand, corresponding to the 3 cohorts. And in each cohort, we practice different harvesting intensity. So we have every time a control stand and a clear cut stand. And then in the young stand and medium stand, we practice 2 different types of partial cut. So before harvesting, in each experiment style units, there were 5 circular permanence plots who were established to follow everything about the stand composition and evolution. So 20 years later, we go on the same plots, and we took measure of temperature, soil humidity, tree density, and deadwood. And for bryophytes, we collect samples by microhabitat using the transect method in each plot. Then we identify them. So in total, we found 71 species of bryophytes, which were advertised in all the treatments in all the stands. And the most of the species were Acrocarpus, Pleurocarpus, or liverworts. So Acrocarpus and Pleurocarpus constitute the phylum mosses, and liverworts are another phylum. So we decided to analyze them separately. And because we didn't observe general pattern in community composition after treatment, like you can see here, we also decided to divide the 3 cohorts and analyze them separately. So to look at the response of bryophytes to treatment in the cohort, we just practice model comparison, and we look at the bryophyte richness, the total, and by life form. And then to look more about the relationship between the bryophyte and the environmental condition, we make model selection. So we test with all the variable measures. We test the model about the climate, the stand, microhabitat, and we also test the combined model, complete model, and everything like that. So what we observe about the bryophyte community response to harvesting. So in youngest forest, so the cohort 1, the total bryophyte richness, including all the life form from clear-cut stand, are really significantly differently from the control. We can see that there is less species found, and this difference was a lot of driven by the Pleurocarpus group. When we look in the cohort 2, we found an average more species in the community from control stand compared to all those who were cuts. And this tendency is particularly visible for the 3 different life forms, if we look at Acrocarpus, Pleurocarpus, and liverworts. But we also found some differences between the partial cut and the control stand, depending on the life form we observe. So this means that the community in hardwood stand were more resilient to partial cuts, and this greater resilience can come from a greater tolerance to changing environment conditions. So we suppose that because in Youngstown, there is a lot of Trembling aspen, an hardwood species, who lost its foliage during the winter. So we suppose that the community is already habituated at the seasonal fluctuation in the stand, like the wind, temperature, humidity, and the abundance of litter. And in contrast, in mixed stand, there is a relatively dense overstory because the conifer species retains the needle all the year. So we suppose that this community are more susceptible when we touch at the stand. And so finally, what's happened in the third cohort? So the results were really different from those we were expected 20 years after harvest. The total species richness was similar between the control and the clear cuts, but we observed only a significant difference in terms of liveworts, which are considered like a really sensitive group. So we know that there is an epidemic of spruce birdworm some 50 years ago. And like this species attacks mainly the basal film and spruce, which provide, like I say before, the protective dome from the understory. But because the spruce bottom maybe make defoliation of the species, we know that this species will die, and then the hardwood species will come to establish, and these altered landscapes leads to more successional stage in the understory. So maybe that's why this community was already habituated to the fluctuational variation in the understory, and that's why we didn't observe any differences. So the stand structure and composition must have a really strong influence on bryophyte richness, and this can be reflected by the model selection. So except for Pleurocarpus species, where no variable explain this group, we can see that for Acrocarpus and liverworts, the best model was always the stand or the microhabitat. So the stand was characterized by the hardwood and coniferous basal area, and the microhabitat by the quantity of dead wood, stacks, stems, and everything. So this variable directly represents the structure and composition of the stand, and this is really necessary to be there. It's necessary to take them into account to practice the ecosystem forest management. So to conclude, the post-harvest biodiversity depends on several factors, so including the initial stand structure and composition, and the rates and spatial pattern of harvesting. So the bryophyte response to the different harvest intensity were not similar among the three cohorts type, but in general, we observe more difference between communities from the control and clear-cut stand than between control and partial cut stand. So this is really a good sign, because since harvesting impact on biodiversity will be minimized with the partial cuts. So if properly applied, we can suppose that the ecosystem-based management can combine the productivity we have with the clear cuts, but also the conservation we have with the partial cuts. But of course, several species were still negatively affected, even when we practice a low harvesting intensity. So partial cuts in all the range of variability, the spatial pattern, and the degree of harvesting, we really need to further study to see all their potential roles in maintaining the species in managed land stands. So I would like to thank everybody who helped me on the field, on the lab, on the right, and everybody who also permits to be here. Thank you.
Thanks everyone for coming to learn about some results on biodiversity from the Jay Creek study. I'm just going to go ahead and thank right now all the other contributors to the work at Jay Creek over the years, some of whom are mentioned here. So an alternative title for this talk could have been, well, how do I fit almost 30 years of data into 15 minutes? So I tried to pick out the most interesting examples, or the ones that are maybe going to spur interesting discussion about what is recovery, how do we measure it, what is the baseline, that sort of thing. Oh shoot, my slides are not moving. Oh, good. Okay. So the Day Creek Silvicultural System Study is located in the interior temperate rainforest of northwestern British Columbia. These forests are dominated by western hemlock and western red cedar, but also have components of 7 other species, 3 deciduous trees, and 4 other conifers, all of which can establish and mix stands after fire. And that is the major stand replacing disturbance. However, due to the cool, moist climate, fire return intervals are not very frequent. And stands over 300 years are not uncommon in the landscape, like this one in the photo. The stands in the experiment were mostly 140 years old when they were harvested 30 years ago. There was also a block of old growth stands that was 350 plus. The Day Creek study is also on the territory of the Gitxsan Nation and the Lahip of Samoyed Mouse, Sikalahan. And I'm speaking to you from the territory of the Wet'suwet'en Nation, home of the Ginundum clan in Smithers, BC. So we'll see if this works. Oh, shoot, it's not working. So we have a virtual tour of the research forest, which shows you the treatments that were put in 30 years ago. And I'm going to just...
I think you need to click the play button.
Okay, it's going on mine now. I don't know if you guys can see it though. No, okay, I'm gonna give up on that idea. I did test it on someone else, but oh well. So we'll just have the static image. We had a unharvested control, a clear cut, and then 2 levels of partial cutting. Can you guys see that? Yeah, okay. So the heavy removal was 60% of the volume was removed in patch cuts that were up to about a ½ hectare in size, and then single tree removal between the patches. And then a light removal where 30% of the volume was removed, mostly in small gaps in single tree removal. And then researchers have been following several different forest values over time: stand structure, coarse weedy debris, small mammals, birds, and understory vegetation. And I do want to point out that compared to some of the other intensive practices mentioned in other talks, where clear cuts are managed for single species with herbicide treatment and perhaps also fertilizer along the way, these clear cuts are actually now the most diverse treatment in terms of tree species, because in addition to the planted conifers, the 3 deciduous species have naturally regenerated. So they're quite nice, diverse young forests at this point. So some of these questions about recovery we've kind of struggled with, and one of the definitions that seems to be simple and people use a lot is recovery as returning to the condition of an unimpacted benchmark system, which seems to work for some of the organisms we've measured. We can compare their abundance to the unharvested forest as a baseline and determine if they've returned to the baseline. Although this seems to work better for partial cuts than, or for clear cuts than for partial cuts, because those are stand replacing disturbance, which is how the unharvested forests were initiated. In the partial cuts, they manipulate the stand in a different way, and organisms may not be on a trajectory back to the unharvested forest. We've created an uneven age stand structure and different new habitat features than existed before. And that might not have existed for another 100 years of succession and multiple natural partial disturbances. So recovery is progressing through normal successional processes in the absence of degradation seems to fit better for a lot of the organisms we've looked at. I've tried to make sense and group some of these recovery patterns by species. So one group is early cereal species with rapid recovery. Another's forest species with rapid recovery. A third is old growth associated species. And those might fit okay in the first definition, but a lot of other things are just more complicated. Yeah, so this is the easiest group for me to wrap my head around, where these are early serious species that come in, but then they have rapid recovery back to the undisturbed condition, which means they disappear once the trees reach canopy closure. So as long as normal succession process is occurring, like the forest is regenerating, then there's no extra action needed for recovery. So early cereal herbs are a really good example of this, where there's hardly any in the forest before disturbance. Then they come in, they like that new open environment. So in all these figures, we're looking at the abundance over time. The red, so it's easy to remember, is the clear cut. The heavy removal is orange. The light removal is yellow, and the uncut is green. And these, by year 24, the early cereal herbs were pretty much back to almost nothing. So we could call that recovery. Early cereal mosses were much less abundant, but they still have that same pattern. They came in after disturbance, but after the canopy was closing, they were pretty much gone. Here's there's two bird species that had this rapid response to recovery. I'll show, this is the chipping sparrow. So it had a peak in abundance around year 10, and then it declined quite quickly as the trees regenerated. It wasn't quite back to the unharvested at year 24, but it certainly looked like it was on that trajectory to recovery. The American robin had a peak earlier than the chipping sparrow, and it declined sooner as well. So last year, we just remeasured our small mammals by live trapping, and meadow voles were another species that rapidly rose in abundance after harvest, but didn't stick around. And we captured very few of them last summer. So anyway, this is like, you know, fireweed, dandelions, voles, and robins, not exactly the poster children for biodiversity. And these early cereal species, usually humans create a lot of the habitat they like. We're not really worried about them. We know this early cereal stage supports a variety of other species, and there's all the invisible species that are harder to measure, like that Terry presented about. So we know this stage is really important, and it seems to be it's an easier one to understand the dynamics, which is great. This other group is forest species that rapidly recover. So they have negative response to disturbance, but then they come back pretty quickly as the shading comes in and it loses that early cereal stage. So feather mosses as a group showed this kind of pattern. They decreased with more harvest. They increase their abundance over time, and it looks like as the forest becomes darker, those feather mosses are going to be probably back to the undisturbed condition pretty soon. Now, this doesn't account for that the mid-cereal stage will be coming along, and perhaps the light environment is actually going to be too dark for these feather mosses, so they're actually going to do another decline, which is another part of why it's hard to define this recovery stuff, as succession is constantly moving. So a bird species, the chestnut-backed chickadee, had this decline after harvest back to the unharvested forest, and actually more than the unharvested forest. So is that better than recovery? And then there's this other group, old growth associates. So these are like the forest dependent species that have really, really slow recovery. They're on the far other side of the recovery speed spectrum. So example at Jay Creek that we've been looking at is the Cyanoligans. They might be completely eliminated by a stand after harvest, especially if they're epiphytes and they need the tree to grow on. And their recovery is on the scale of centuries. So in BC's North Coast, we have data on epiphytes that shows it takes over 250 years after standing replacing disturbance to get the same community back. I think the #1 management action for these kinds of species is conservation planning. However, there's reason to experiment with silviculture that emulates those smaller scale disturbances. For species like these Cyanoligans, they're limited by microclimate and dispersal distance. Smaller openings and shorter distance to edges may maintain them and allow us to still extract timber. So here's a species of blue-listed lichen, smoker's lung lichen, where we've finding them colonizing small openings on 25-year-old regenerating trees, but mostly close to the forest edge. So this figure shows the colonies we're finding per tree as we go farther from the edge, and these sensitive species we're finding mostly in small openings and closest to the edge. So I've lost a lot of time with my technical glitches. There's all these other species that I talked about that are just really more complicated. I'm going to just quickly go through some of these. Birds loved the heavy removal. That orange line, they came in and doubled their abundance in this treatment. And there was about 15% more species. So we think that is related to the pattern of harvest, where we've created these gap cuts and a lot of strong edges. The birds, a bunch of birds from a variety of habitat groups seem to just love these edges. So my takeaway here is that, well, harvesting that creates a lot of edges may be desirable habitat for a wide range of birds. But of course, succession softens those edges and that effect declines over time. So if we want to keep that higher level of bird abundance and diversity, we'd have to continue to do these gap cuts. So is this recovery? And then we're seeing how the shrub specialist birds follow the deciduous shrubs so closely. So if we want more shrub specialist birds, we just need to grow shrubs. And similarly, our snag specialist birds were associated with more deciduous snags. So here's another example, a young conifer lover, the magnolia warbler. So there was hardly any magnolia warblers at the initial at Date Creek, but by year 24, where we have this young conifer component in all of the stands now, there's now tons of magnolia warblers. This doesn't look like recovery. The abundance is actually getting farther from the benchmark condition over time. There's a new component of stand structure which supports this new bird species. And we may have a young conifer here. We just had these results this week from our deer mice. And unlike the magnolia warbler, which like those treatments with lots of young conifers, the deer mice are much less abundant in these treatments with all these small conifers. So maybe we're being premature and we don't know, everything that these mice are responding to. But the benchmark of definition of recovery doesn't seem to work for a lot of these species. And what seems to be more important is keeping the habitat features around that they need to have, and to keep those for the retention and plan for continued recruitment of that habitat over time. Am I out of time?
Yes, I think maybe you can present maybe up to 1 minute.
Okay, so the recruitment of, that brings me to this slide about coarse woody debris, because it's one of those habitat features that needs retention and recruitment, and hosts all this other invisible biodiversity that we haven't comprehensively monitored at Date Creek, specifically the fungi and arthropods. So we're hoping to, use some of this coarse woody debris information to just as this coarse way to capture a lot of these other species. And we're seeing that the coarse woody debris increases over time where we've left retention, where there are trees to die and blow or be blown over live. The main contributor of new fresh woody to pieces is windthrow, which is this bottom figure. So just because I'm very short on time now, this is just a visual that kind of brings that all together. This is on the left how a complex pile of coarse woody debris develops over 29 years from windthrow, where a gap has continually fresh pieces falling in, and a 29-year-old slash pile, which after 29 years is not supporting the same kind of habitat value. Okay, so we'll call it there and hopefully come back to some of the stuff in the discussion.
Okay, thank you, Erica. So we have to move on to the next speaker, Paul Fornworth. Okay, so Dr. Fornworth is a research ecologist working for the USDA Forest Service Rocky Mountain Research Station in Colorado. So today she's going to present understory plant responses to salvage logging management alternatives in beetle affected lodgepole pine forest. So please welcome Paula.
Hi, everybody. Can you hear me? All right, great. Thank you. So yeah, thanks for the invitation today. I'm going to be talking about some work that my colleagues and I have been doing looking at understory plant responses to salvage logging management alternatives, which I'll explain what that means, in beetle affected lodgepole pine forests. So the mountain pine beetle outbreak that has affected over 1 million hectares of lodgepole pine forests in Colorado, and as you all know, millions more hectares across Western North America, is in the rearview mirror now. In Colorado, beetle activity largely occurred from 1996 to 2011. And in Colorado, this outbreak was often quite severe. We had 80 to 90% of lodgepole pine basal area killed in many stands across the state, and 60 to 90% of the total basal area killed. And so the action or the do something management response generally involved salvage logging, or more specifically, the removal of dead and dying lodgepole pine trees. And these salvage logging treatments were conducted over a relatively small portion of the infested stands. I think that's important to note. And they were done primarily to capture the economic value of that dead wood, and then also to address fuel, water, tree regeneration, and other concerns. But as you all likely know, there's more than one way to salvage log. These are the management alternatives that I was referring to in my title. And so the management alternative that a manager might choose depends in large part on what their objectives are, right? So if their objective is to reduce elevated fuel loads, then they might choose to whole tree harvest, where in addition to removing the merchantable material, the slash is removed from the site. If they're interested in helping to retain snow on site and increase water delivery, then they might implement a bowl only harvest where the merchantable material is removed, but the slash is retained on site. It's lopped and scattered on site. And if they're interested in regenerating that next forest, then they might follow a whole tree harvest treatment with a mechanical scarification treatment to prepare the seed bed. And so today I'm going to discuss some research that is evaluating the effects of these three contrasting alternatives on understory plants, specifically tree regeneration, and then also the graminoid forb and shrub community. And some of these results have been published already in these two relatively recent articles in Forest Ecology Management, and some of the results are preliminary. So our work was conducted in 4 study areas in northern Colorado. And in these study areas, lodgepole pine attacked mountain pine beetle beginning in the late 1990s to the early 2000s. And then the sites were salvage logged by managers, national forests, and also state forest agencies in the winter of 2008 and 2009. And by and large, what they did was whole tree harvest, where the dead and dying trees were cut, and then depending on the management agency, the live trees were either also cut or they were retained on site. And so what we did is we went into these 4 study areas and we established 3 replicates of an experiment. So that gives us 12 blocks of our experiment of these different management alternatives. So our experimental treatments included the uncut treatment, so the no action alternative, and it included 3 action alternatives. So that is the whole tree treatment, the bowl only treatment where the slash was brought back to the site. Remember, these sites were operationally whole tree harvested. So we returned the slash to the site, and then the scarify treatment where we went in with a dozer and scarified the soil. And so first I'll talk about tree regeneration. I'll just talk about this pretty quickly here. This is my only slide on tree regeneration. And it's a pretty complicated graph here. You'll see we have the 3 different panels reflecting 3 different age or size classes of regen. So this top panel is our new seedlings that established after the logging was conducted, and then 2 size classes of advanced regeneration that had established prior to the logging, but sometimes in a lot of cases actually after the mountain pine beetle. And so just hitting some high notes here with regard to lodgepole, that's this first column. I don't know if you can see my cursor. This first column of species is lodgepole, and it was highest in the whole tree treatment, and intermediate in the other 2 logging alternative treatments, and lowest in the uncut treatment. And this pattern kind of followed for the other 2 size classes too, but you really see it in this, what I call the new recruit stage or the new seedling stage. So bell pine fir, which is the next species over, was much less sensitive, if you will, to the treatments. But, you know, generally speaking, if you kind of squint, the uncut treatments tended to have a little bit more subalpine fir than the other treatments. And certainly when you look at it on a proportional level. If we jump over to aspen, which is the last column here, we see that the Scarify treatment really stimulated aspen suckering. It was highest in that treatment. And then also talking about the Scarify treatment, if you were to basically sum up across all these species and look at them, you would see that while the scarification treatment, you know, it may or may not have more regen in it than the bull only and the whole tree harvest treatments. Basically, we saw ample regeneration in those other 2 harvest treatments. And so the scarification itself wasn't necessary to stimulate the regeneration. So moving on to the graminoid, forb, and shrub communities. So this first figure here is of total cover. We sampled 2 years following treatments, and then again 9 years following treatments, and we have our 4 treatments across the x-axis here. And so first, if we compare the 3 action alternatives to the uncut condition, we see that generally speaking, total cover can be robust to logging. But then if we look just within the 3 action alternatives, we see a logging gradient, where the Scarify treatment, which I think of as the most aggressive disturbance, it reduced the cover relative to the whole tree treatment, which was kind of the de facto treatment, right? That's what the managers tended to do. And then the bull only relative to the whole tree encourages it. Again, significances can differ, but basically it's this gradient, and you see it in both years, this gradient. And so I think something that's interesting is looking at our soil data. It provides insight, I think, into why we might be seeing that bull-only treatment having greater cover relative to the other management action alternatives. And this first column here shows soil moisture, and you see that that bull-only treatment had greater soil moisture than the other 2 logging treatments. And then if you look, these next 3 columns here, if you can see my cursor again, this is plant available nitrogen. And if we focus just for ease on this last column, which is the total plant available nitrogen, again, it was higher in the bull only treatment than in the other 2 logging treatments, and also higher than in the uncut treatment. So I think that these soil resources may be explaining, at least in part, what we're seeing specifically with cover in the bull only treatment. So in addition to looking at totals, we looked at things by functional groups. And so the graminoid and the forb cover was robust to logging, but the shrub cover was not necessarily. I think the main thing that you see here again is this gradient, with shrub cover being greatest in the bull only treatment and lowest in the Scarify treatment for both time periods with respect to the action alternatives. And so this is just a picture here to show you what one of our uncut stands would look like. And the shrub that's really dominating our communities is this low-growing vaccinium species, mostly Vaccinium scoparium, but that's not the only species that we had. And this is a closed forest species that's very sensitive to exposure. And so it's likely that the bull-only treatment may have provided Vaccinium with some protection. So maybe not necessarily water, although I'm sure that didn't help, but perhaps shade. We did not quantify light. So moving on now to total richness, it also can be robust to logging. Again, we see this gradient, but it's inverted with total richness being greatest in the Scarify treatment and lowest in the bull-only treatment across the 3 different management alternatives. But again, generally robust because differences between the 3 logging alternatives with the uncut were not apparent in either sample period. And so again, looking at functional groups, it's a lot of the same thing. We see that we're seeing those, where we're seeing increases in richness, it's due to increases in forb richness, as well as in graminoid richness. And so that's it. That's all that I had to share in terms of data. In terms of implications for recovery, because I know that we want to have a discussion, especially about longer term implications for recovery. What I think kind of the key take home of our project is that recovery is affected by decisions about whether you log and then how you log in these beetle affected stands. And so with regard to trees and future forest conditions, the 3 logging alternatives may be the best ones if what we want to do is return stands to lodgepole pine dominance, which is, what they were before the outbreak came through. If we stick with the uncut alternative, then that may advance the stands at least more quickly towards subalpine fir dominance. Not necessarily, again, because we had more subalpine fir, but mostly just because of the proportional lower abundance of lodgepole coming in. And then with regard to graminoids, forbs, and shrubs, total cover appeared to be fairly robust, though we did see shifts in composition. We saw the gradient with that bull-only treatment potentially encouraging cover the most, or the recovery of cover due to greater soil moisture and nitrogen, and then possibly also light. And then we saw shifts due to logging from a more shrub-dominated community to one that had more of those herbaceous forbs and graminoids. And then also that total richness can be robust to logging. Again, with that Scarify treatment, the most disturbance, the treatment that brought the most disturbance, increasing richness the most, and the bull-only treatment having the lower richness among the management alternatives or the action alternatives. And in contrast to the shrubs driving the cover response with regard to richness, it's largely due to what's going on in the forb and graminoid communities. And so that's it for me. Lots of people to thank as always, right? So I'll pause there.
So something happened where I ended up losing my first slide suddenly here. So this is, I'll just go with that. So thanks for the introduction, Seung Il, and my name is Eli Swanson. I'm a postdoctoral scholar at Oregon State University, and unfortunately, because my first slide isn't up, it doesn't show my co-authors on this presentation, nor my title, but I just want to acknowledge my co-authors, Dr. Klaus Putman, also at OSU, and Dave W. Peterson at the Pacific Northwest Research Station in Wenatchee. And so my presentation, it's a little bit different than the previous ones in this session in that what we really want to present is an idea or a concept paper that we are developing right now. And so if any of you have ever listened to Klaus's presentations, you're probably familiar with his phrase, before the fire is after the fire. And so it's kind of this funny, what sounds like a simple logic puzzle, but really when we think about recovery of ecosystems and developing restoration tools. So I'm a restoration ecologist, and I think a lot of times about how to put ecological theory into practice in a restoration context. So when we think about developing these tools, we really want to think when we have a disturbance like wildfire, how can we plan for the next wildfire? So the primary objective of our study was to develop a model and a set of general principles based on key ecological processes and functions to guide pre- and post-fire management aimed at restoring adaptive resilience across multiple forest types. So, one of what we really focus on a lot in the research that I do with Klaus and Dave is dry forest systems, but this model and principles is really applicable to multiple forest types and multiple disturbances. So this objective stemmed from the 2020 Forest Service Ecosystem Restoration Policy, and that policy gave broad guidance for restoring ecosystems on national forest system lands, so that if they are subject to disturbance or environmental change, they have the ability to reorganize and renew themselves. And so those last two terms are really key as we encounter more disturbance in the Pacific Northwest, and we have more frequent fires. Reorganizing and renewal of those forest systems is going to become more and more important, primarily because I'll share this burned area emergency response map. So primarily those terms, renewal and reorganization, are important because with these high severity fires, we end up with such a large area burned that active restoration just becomes impractical. And so what I really wanted to highlight in this bear map is, if you see all these areas in dark red, those are areas with over 90% basal area mortality, so really high severity. And just, this was a very large fire, and there's just far too much land area to actively restore. And so active restoration, what I mean by that is people are on the ground, actually they're applying restoration by hand. And so we really need a different approach for forests to be able to renew themselves. As restoration ecologists, we want to try to provide the building blocks for this to occur. So what are those building blocks? And let's see if my slides advance. So that's really going to depend to some degree on the forest type and condition. But there's three general themes with research-based evidence for supporting resilience. And so what I did was a systematic review of over 300 studies, and this is just a very small sampling of those studies. But many of us are likely familiar with C.S. Holling's work on developing concepts within resilience, and for the intents and purposes in our work, we're particularly interested in resilience as explained in his more contemporary work with Lance Gunderson, that really incorporates adaptivity and reorganization after disturbance into how they are defining resilience. So these earlier works by Holling and Gunderson really provided that foundation to confront this sort of current era of very dynamic ecology and global change that we have all found ourselves in. So these general themes have been referred to in the literature sometimes you may have seen them referred to as resilience assets. So anything in this loop diagram with a plus sign is indicative of a resilience asset. So first, in the upper left, we have foundational structuring processes, and there are 4 key processes evidenced to build resilience that can be actually incorporated into active restoration goals. So dispersal, natural disturbance regimes, recruitment, and reproduction. And then resilience can be further constructed by incorporating functional drivers that support those processes. So functional connectivity, functional response diversity, functional redundancy through pattern and response. And then another key piece of resilience that we want to highlight is ecological memory. So while we can't restore informational legacies after wildfire, we can work to protect and conserve them before fire occurs as part of a risk mitigation strategy or avoidance. And we can mimic material legacies through restoration if fire is severe enough to destroy them. So while we have resilience assets, it's also important to note that there are certain elements that erode resilience, and we can call those resilience debts. So altered disturbance regimes, invasion, and climate change. So all of those resilience debt, they can interact and exacerbate negative effects through positive feedback loops. So while these debts erode forest resilience, they also limit restoration success, and understanding how can help us design restoration programs that simultaneously deconstruct barriers while also building broader resilience. So if we set aside those resilience debts and we just focus primarily on the assets and those assets that will help us operationalize resilience, we have developed 3 general principles that managers can consider when designing restoration programs for their forests. These principles are general enough to guide the development of restoration objectives across many different forest types and are applicable to both pre-fire and post-fire work. And so in this session, we're really focused on recovery, but we think that there's sort of this gap between, what happens in restoration before fire and restoration after fire. And so making principles that managers can use in both contexts might be useful. So for our first principle, we suggest identifying legacies that support processes and functions so that those legacies are essentially inventoried and accounted for, and managers can utilize that information, those informational and material legacies that will provide continuity between pre-disturbance and post-disturbance landscapes. Second, integrate and process refugia in pre-fire stand restoration plans by identifying conditions that create refugia, or in post-fire environments, identify and protect disturbance refugia. So disturbance refugia in many forms and sizes increases resilience, connectivity among forest patches, and adaptive capacity after fire. Lastly, we want to inventory and evaluate pre and post-fire functional heterogeneity, functional connectivity, and redundancy, and really develop those where they're lacking. But this is all theoretical, and designing restoration programs that build resilience is actually really challenging. Especially, I'm sure anybody that has done restoration activities in the field probably knows that this is very challenging. So because many of the types of forests, there's many different types of forests in the Western United States, this necessitates a flexible and adaptive approach that can be applied across, the vast array of forests that we have here. So here's our general conceptual model for this proposed approach to restoration, and we're calling this the complex adaptive systems resilience, or CASAR model. And so again, and anyone that knows Klaus knows that he does a lot of work in complex system science, and so we really drew deeply from that to create this approach here. These boxes that you see here, we are calling those fire adapted restoration packages. And so with a starting goal of restoring forest resilience before and after fire, we can design these FAR packages that deliver ecological processes and function to restoration sites depending on that site condition and forest type. So the manager would then use the aforementioned principles to build these packages by adding relevant legacy information, identifying processes and drivers that support resilience given their site conditions. So the goal here is to restore pre-fire forests to capacity where when wildfire occurs, that forest has the pieces in place where it has a chance to reorganize and renew, and only passive intervention is then needed, which would hopefully reduce the amount of resources needed for restoration. Alternatively, in post-fire forests that haven't been previously restored, active restoration is used in a similar manner to rehabilitate the site with keeping in mind the possibility of another wildfire and the goal of reorganization and self-renewal, which, if we want biodiversity recovery after disturbance, really it's very similar. We want to have those building blocks in place. So if we take a closer look at one of the FAR packages with an example from the Holiday Farm Fire, and the Holiday Farm Fire was the fire that I showed you in that burned area emergency response map. So at this site, again, we had over 90% basal area mortality, and this photo was taken about a year after post-fire, and you can see some maples popping up in the foreground, but prior we had a pretty blank slate. So first, the manager would determine the goals for the forest based on the site conditions, and these are placed on the axes. I know I'm getting close to time here, so I'll just do this really briefly. I just wanted to introduce one of these packages. And then I know this site, so I know that it has a slightly south aspect and will likely encounter some environmental stress that limits recruitment. The burned area is very large, so likely also dispersal barriers. So once our axes are set and the barriers are identified, then we can get into the restoration practices that are actually applied. I won't get into detail on that, and but I will just end by sharing my contact information if anybody has any questions they want to talk with me about, and acknowledging the folks that provided me with the photos for this. Thank you.
Thanks, everyone. Thanks for staying till the end. I'd like to acknowledge my co-author, Chrissia Tabakaru, who is not able to be with us today. I'm with Alberta's Provincial Forest Agency, and Chrissia is with the Alberta Biodiversity Monitoring Institute. I'll be presenting some of ABMI's work as well, but just be aware that ABMI is independent from government. I also want to acknowledge Jennifer Hurd at ABMI, who has contributed to this work as well. What I hope to give to you in our talk is a perspective of how understanding the recovery of biodiversity following forest harvesting feeds into strategic and operational planning in Alberta. So Alberta's landscape is a busy place with multiple overlapping natural and human disturbances, including fire, harvesting, and oil and gas. Addressing the cumulative impacts of these developments is important for maintaining ecological, social, and economic values that these landscapes provide. So Alberta is therefore developing an integrated land planning system called the Land Use Framework, which has established 7 land use regions and which calls for the development of a regional plan for each. So far, plans have been completed for 2 regions, including strategic objectives and the establishment of new protected areas. These regional plans will provide direction to sub-regional and operational level planning through environmental management frameworks. Environmental management frameworks are a key part of this cumulative effects management system by setting triggers for key indicators. EMFs are being developed to address air quality, different aspects of water quantity and quality, and to address biodiversity. And I'll focus in a minute on the biodiversity management framework. However, I will note right now the current importance of caribou range plans, as they have taken on a recent significance that has complicated this planning hierarchy. These caribou range plans are a special kind of sub-regional plan that is a major initiative in Alberta right now, with 2 plans finished and many more underway. These plans are driven by federal provincial agreements to address species at risk requirements. What is relevant for our discussion today is federal direction to set disturbance limits in caribou ranges at 35%. The disturbance recovery is therefore an important issue for forest planners. The forest sector in Alberta is very interested in knowing what silvicultural systems can be used to promote recovery of disturbance from a caribou perspective. But now I will discuss the biodiversity management framework, as its development has also led to much discussion about post-harvest recovery of biodiversity. The current vision for biodiversity management frameworks is that they will contain habitat and species level indicators for both terrestrial and aquatic ecosystems. Terrestrial habitat indicators in development right now include habitat intactness, interior habitat, and old forests. Triggers will be set for each indicator, which, if crossed, initiates a management response. Trigger levels will be set based on the assessed risk of the current condition. The level of management response will also depend on the risk level to biodiversity shown by the indicator. Here's an example of some work already done. The 2018 status of the interior habitat indicator. The settled agricultural area here has the lowest amounts of interior habitat, and these are the areas you can see here in red and orange. Whereas watersheds in protected areas in the Northeast and in the Rocky Mountains have the highest levels of intactness, interior habitat. And these are areas in blue. Now, a key criteria for each biodiversity indicator is a monitoring system so that it can be measured and tracked. This is why the Alberta government has partnered with the Alberta Biodiversity Monitoring Institute, which tracks changes in Alberta's wildlife and their habitats, and which collects information on thousands of species ranging from mammals, birds, amphibians, invertebrates, and plants. These on-the-ground measurements are complemented by regional to provincial level mapping information products. For the terrestrial habitat indicators I mentioned earlier, a particularly important product is the human footprint inventory. This inventory was created through a partnership between the ABMI and the Alberta government agencies. And the human footprint status and trends are monitored using high-resolution satellite data and through annually updated inventories. This inventory is a comprehensive, up-to-date digital representation of surficial human disturbances across Alberta. Feature ages range from a year to multiple decades, going back to the 1920s for forest harvests, and includes temporary, successional, and permanent features, 115 types and 21 categories, ranging from reservoirs to roads to mine sites, urban features, cultivation, and forestry. However, it's important to understand that all features in this inventory are treated equally, and that succession or reclamation of human footprint is not modeled. The dynamic status of temporary features, such as harvest blocks, has not been reflected in this human footprint inventory dataset. HFI is a cumulative dataset. So this inventory provides a solid baseline for biodiversity monitoring. However, for the purposes of strategic and operational planning, this dynamic status is important. This is important to understand when we consider forest management planning. Forest management plans in Alberta are required to plan out 200 years into the future with the next 20 years of harvest spatially laid out. These operational plans need to be consistent with strategic plans, such as the biodiversity management frameworks. For us to be able to assess consistency of an operational plan with higher level plans will require being able to forecast the future condition of BMF indicators based on proposed operations through time. Therefore, future projected human footprint also needs to consider succession and forest recovery following harvest, or else the accumulation of future planned forest footprint would quickly cross biodiversity management thresholds. Therefore, for the biodiversity management frameworks to meaningfully influence forest harvest planning, we need to be able to quantify forest recovery in a forest recovery curve. So what are some of the basic parameters of this curve that we need our research partners to quantify? In describing the parameters of a recovery curve, I'll show graphs with years after harvest on the x-axis and percent pre-harvest on the y-axis, or pre-harvest biodiversity on the y-axis. Just be aware that the y-axis is the inverse of human footprint. So for example, 0 footprint would equal 100% biodiversity. Okay, so the first parameter we need to know is, what is the impact on biodiversity at the time of harvest? Does it go to 0? Well, we don't think that's likely, as forest harvesting while altering habitat doesn't eliminate it. So to what level then? We also need to know whether biodiversity fully recovers after a period of time, or whether it only partially recovers to a certain level. From work done to date, we are now assuming full recovery. Another parameter we need to know is how long does it take to get to full recovery? 80 years is a number we are currently looking at, but could it be longer or shorter? Finally, what is the shape of the recovery curve? Does recovery start off slowly and speed up toward the end? Or is there a quick initial rate of recovery followed by a slow final progression towards full recovery? Selecting a curve is not just interesting ecologically, but identifying each of these parameters that define the recovery curve has real-world operational impacts on the level of harvest that can be sustained below a selected biodiversity management framework indicator trigger. The need to develop a recovery curve with the appropriate parameters has led to several initiatives. The first one I'll discuss is an ABMI project which utilizes satellite remote sensing of vegetation. Oversimplifying greatly, this project takes advantage of changes in the normalized burn ratio index from forest vegetation as detected by Landsat. Here we see a sequence of Landsat images of a cut block from 1985 through to 2017. Skipping the rest of the methodology, the end result is the harvest area spectral regeneration product of 2018, which contains spectral regeneration information for almost 58,000 harvested area polygons, using a Landsat time series over 34 years. In this product, harvest areas were 60% spectrally regenerated after 5 years and 80% regenerated after 8.7 years. Overall, 68% of the harvest areas in the dataset have reached 100% spectral regeneration. This is an amazing product, but there are a few things that need to be considered. Spectral regeneration measures light reflectance signals across a pixel over time, and therefore doesn't directly equate to ecologically ground-based definitions of regeneration or recovery. One important limitation is that the spectral responses eventually stop changing as the forest grows due to a saturation point reached after 15 to 20 years. Therefore, useful as this product is for many purposes, we are pursuing other approaches as well to determine the recovery curve of human footprint after harvesting. So over the last few years, the government of Alberta has collaborated with the ABMI on a few projects. In 2015, our partnership commissioned a literature review and meta-analysis that in the end involved 17 studies and ABMI data. This analysis went beyond just looking at measures of forest structure, but included direct measures of biodiversity, such as post-harvest trends in vegetation, birds, invertebrates, and mammals. The analysis showed that in deciduous stands, it took 24 years to achieve 50% recovery, whereas in conifer stands, it took 42 years. One issue with this analysis that generated a lot of discussion is that the analysts scaled the recovery curves to 0 at the time of harvest. This was challenged by some. There was also a desire to go deeper with this question, to include more data sets and to investigate more approaches to recovery curve development. This led us to approaching Ellen McDonald at the University of Alberta to address our questions. The first phase of this project focused on identifying suitable methodologies and data sets for the development of a post-harvest recovery curve. It was led by Ellen, Samuel Bartels, and Scott Nielsen. Indicators of recovery were assessed, such as univariate predictors, including species richness and abundance, and multivariate approaches, such as species composition. Methods to assess time to recovery were also assessed, such as the ordination regression-based approach and relative recovery approaches. They also conducted a systematic literature review to identify relevant data sources. Based on the recommendations and findings from phase 1, phase 2 is initiated to actually develop the recovery curve itself. This phase is still in progress. This phase is led by Anne McIntosh, Ellen McDonald, Singil Lee, and Selena Shute. Anne McIntosh and Selena Shute presented part of this work earlier in their session, in their talk on EMEND and with other data sets and recovery after variable retention harvesting. Currently, this study is collecting and analyzing data sets from other post-harvest studies. So I'll conclude with just a few questions from a forest management perspective. The first question is, what is the relationship of stand structural metrics to direct measures of biodiversity? I asked this to try to find a connection with forest planning indicators to direct biodiversity indicators, so that forest planning can better manage for biodiversity. The second question relates to identifying silvicultural practices that speed up the recovery of biodiversity. And finally, a question for us as forest managers: can we provide incentives in our forest management system to promote implementation of practices that speed up recovery? With that, I thank you for listening, and here's some contact information, and we welcome questions and discussion. Thank you.